Hey guys, welcome back. So, in this next section of Module 9, we're going to talk about the bacteria. Okay, so I know you've been waiting and you've been wanting to get there, so here we are. Um, a lot of books, biology books, will separate this out a little bit more. It's okay. I think for the intensive purpose of giving you a little bit of a background, a good taste, this does give you a good taste of that. If you want to know more, take a microbiology class, and you'll learn a lot about bacteria. I loved my microbiology class. There's several that you can take in college, so just wonderful. One of the things I want you guys, I love in the Pangea book is things to ponder. These things you don't get in a secular biology book just because I don't think they really want you to think about this. But this is some things to ponder because our God's pretty cool in that He made all this little life that we don't get to see, and even with our microscopes in class, we're not going to get to see them. Um, just saying, we might get to see, if we got 1,000x, we might get to see some, um, but this is not something we get to enjoy all every day, all the time. But our God gets to enjoy them all the time, and He created them, and He knows them, and He. That's what's really cool. So here's some things to ponder. It says the combined weight of all the microorganisms far exceed the combined weight of all living organisms on the earth. Okay, so all the microscopic life far outweighs all the macro life, all the life that is huge. All right, that just blows my mind. So if you could put it all in a pile and weigh it, it just blows my mind. But we can't do that. But God can. I mean, that's what's cool. This is another thing to think about. So the number of organisms from bacteria, the bacteria domain on our body, your body too, exceeds the number of cells that make up your body. Okay, so we have trillion of cells, and they outnumber your cells 10 to 1. So for every cell you have 10 bacteria on your body. Okay, no wonder we itch, right? Okay, no wonder we got itches and we need to scratch, which means they make up 1 to 3% of your body mass. So if you weigh 100 pounds, that's anywhere from 1 pound to 3 pounds of bacteria. All right, so you can say that 3% of your body weight or your body mass is from bacteria, so you can't lose that. Okay, which by the way, we do. When I was teaching secular biology, we did talk about you could scrub your skin all the time, and you're probably getting rid of some of them, but they're going to come back. Um, and we're going to learn about some of them that they're basically inert. They're not going to not inert, but basically they're not going to mess with you. They're not going to be a pathogen again, unless there's opportunity that allows for them to be one. And again, most organisms are opportunists. They're looking for opportunity because they got to feed or they got to make their own food. So of the 10,000 different, there are 10,000 different microbial species that call us home, humans home. And they play vital roles in our life. And so we're going to talk about that within this section because most everybody goes, oh no, it's a disease, it's strep throat, or it's staph infection, it's MRSA, or I got pneumococcal pneumonia, or I got, just all we talk about is like we think of diseases first and not think of all the good things that they do for us. Okay, so let's talk about them first. We're going to get the ancient guys out of the way because these are the guys that, again, odds of seeing them, odds of dealing with them are not that great because just because um, we don't live in that environment. That's not something where you're going to build your house next to. However, our methogens are one of those ones that's kind of interesting in that they live in an oxygen-free environment of the digestive tracts of cows, and they live in swamps, okay, and marshes, okay. They use hydrogen and carbon dioxide for energy, and they produce methane. And if you guys, I don't know if we really talked about it uh, back in the ecology chapter, but one of the things that they talk about releasing methane into the environment. Well, methane's a bad gas in the out in the atmosphere because it um, traps light, adds to the greenhouse effect, which is not a bad thing because we've talked about that. It keeps our Earth warm, but this could does add to global warming, and so the manufacturing of not manufacturing, but the growing and harvesting of cows. They want us to go back and eat plants and not eat cows because more cows you have, the more methane you have. The more cows you have, the more processing you have, the more waste you have in the environment. So just don't eat cows. Don't eat meat. And so that's where some of that is coming from is right here. Yes, they do produce methane. But you know what? So do your bacteria in your gut produce methane. Trust me. Okay. You've probably smelled it. The other thing I want to mention, and if you lived in Florida, you live near a swamp, you've seen this, where the methane comes up and it's warm and it lights, catches fire, and it is really cool because it makes this light. And growing up as a kid, I had friends that would go and chase the lights, and that's what they were doing. They were chasing the swamp gas, and basically what happens is you end up in the swamp, and you had to call my uncle who had a uh, tow truck service, and he would come and get you and open your car door, and all the water would fall out. So don't chase lights. It's just swamp gas. It could be an unidentified flying object, but it could be just gas. Okay, could just be gas. Um, the halophytes are the guys that love salt, and that's again the Great Salt Lake City, and also too. And I hope this is going to go to the Dead Sea. Um, yeah, so here's the extremophiles in their habitat, and so we talk about the haliophiles, the Dead Sea. So they have bacteria there that live only in that environment, which they're needed there. So don't think that this is something they don't need. So again, Salt Lake City, the Dead Sea. This is where they live. They like that high concentration of salt, um, and they live there. So pretty cool that God made some bacteria live in some very inhospitable environments. Um, the next guys, these thermophile guys, we can't get there. Let me go back. The thermophile guys actually live in the hot springs of Yellowstone, but also in these deep hydrothermal vents. Okay, and in these deep hydrothermal vents is the kind of the basics of uh, the Meg. That if you watch the megalodon shark, the idea is that these vents are down there, and it's real, and it's true. They are there. They are down there. There are critters that live down there in these hydrothermic vents that don't really live any other place in the world. Um, and some of these critters that live in these hydrothermic vents are critters that if we saw them, their brothers and sisters up near the ocean seashore, they'd be like really really small, but they're like 6 foot down there. So pretty pretty cool. Let me see. I'm gonna try to get us to look at some of the vents. But the idea behind the Meg was that these vents, that the Meg lived underneath him in this like environment that he could grow as big as he wanted to grow. Or I'm saying he. Um, they don't have a choice. They're going to eat. They grow. And when they poked a hole through the vent through this area, then that allowed for the Meg to come out. Now I like watching the Meg. I thought it was pretty funny. Um, it's pretty interesting. It's just it's sci-fi. It's not real. Okay, so again, as you watch those things, just kind of keep that in mind. So here is let me see if I can get us. I don't did it go there. Hopefully you can see it. So from here, let's go over here to the. So here is the Woods Hole Oceanographic Institute. They're doing some of the leading research on this, and again, just really really cool. These are tube worms that are living down there. There are critters down there that defy explanation. If you watch any Shark Week and you're watching the one where they're pulling the nets up from the deep dark ocean, and they pull out these critters and go, oh, here's a goblin shark, or oh, here this is make them out. Sharks that they haven't seen before. So again, they found the 6-gill shark, and just all kind of cool things that were down there floating around living. Um, that again we didn't get to enjoy until we sent a submersible down there to go down there and look at them. And we still, as a human being, unless we get in a submersible to go down there, don't really get to see it. So that's pretty cool that God made those critters too that live down in there. So again, these guys are the ancient guys, what I refer to as ancient guys, and we don't tend to. If you see them, awesome. If you smell the effects of them, you might have smelled the effects of them. You might have seen swamp gas. If you've been to Great Salt Lake or the Dead Sea and floated around, maybe you got some ammonia. But they're again, they're not going to cause us trouble, or they're just not ones we're going to be so familiar with. Now the next guys that we talk about, the bacteria, these are the guys that we're familiar with. And for most of the time, like I said, we think of them as a disease. However, lots of them are great. They make vitamin B for us. They make vitamin K for us in our gut. There's very few that are a pathogen, and a pathogen is a disease-causing agent. So it may not be a bacteria that's a pathogen. It could be a virus, or it could be a protist. Protists can be pathogens, or it could be a worm. Worms can be pathogens. So, and we'll talk about that when we get to those guys. So let's talk about the bacteria. Um, Bacteria are pretty interesting, and we've seen some of these words before. Some of them we're getting introduced to. Bacteria kind of have a capsule around them. So, think about when you're taking a vitamin and it's like got a capsule around it. And what does that do for you? What does that capsule allow for that medicine that you're taking? Well, for most part, it allows for it to be time released, or it allows for you to digest it very quickly. Okay, I think the new aspirin that's being um, touted right now, that's being advertised, is got a code on it that doesn't hurt your liver or something. Anyway, it doesn't. It enables it to be digested quickly and not have any side effects from it, not hurt your stomach and stuff like that. So, and we know we didn't talk about bacteria, prokaryotics, as far as having a capsule, but pretty for the most part, bacteria have some kind of capsule. They're encapsulated with something, and we'll look at that here in a minute, and that distinguishes some of them. They got a cell wall. We know that. We talked about that back in our cell chapter, module 4, our module there about cell and cell functions. We talked about that there. So we got a cell wall. This cell wall is not like a plant. It's not like a fungus. It's very different. It has different parts of it. We already learned that the cell wall of this bacteria are very different than the extreme bacteria, the ancient bacteria. We know that everything has a plasma membrane. So of the cell theory, we have a plasma membrane. We have cytoplasm. Every critter, every cell should have cytoplasm, should have DNA. And so for the most part, we've talked about with bacteria that their DNA is in this plasmid and it's circular. And we talked about that in genetics because it's very easy to cut a circle and know where the start and the end is, and to put another gene in there. So we know that. We've already mentioned some of these things. So they got a plasmid. They got these little pili structures, which are kind of like cilia, which we're going to talk about with protists. They kind of allow, again, everything allows for the bacteria to hang around, to do its job, to be in the mix of survival. Okay. And then some of them have flagella, which allow them to move. So this is a very typical bacteria. Now, some of them have more than one flagella. Some don't have any. Some of them have very different cell walls. Some of them have different capsules. Some of them have, believe it or not, I think some of them have more than one plasmid. Some of them have pili, some of them don't. They all have ribosomes, but again, they don't have anything that is membrane bound. So no nucleus, no chloroplast, no mitochondria. So they still got to do all that, but they've got to do it within the functions of their cell, not with any special organelles. Okay. So let's kind of go on and talk about how we're going to identify them. We identify them by what they look like, what shape are they, what their cell wall. We just talked about that. Their cell wall is different, and then how they move. And so when we look at shape, we're going to look at Are they round? Okay, so we're going to look at cocci, diplococci 2, staphylococci looks like. So all your staph infections look like this. How do you know it's MRSA versus the other staph? Then you have to do some other tests. Streptococcus, all our streptococcus looks like this. So your strep throat looks like this. How do we know it's this one and not another one? Again, you have to do more tests on that. And that's when they take a culture, that's what they're doing. They're culturing that bacteria. We have rod-shaped bacteria, and then we have spiral bacteria. And so we kind of look at those two. So as we look at them, here's a bacillus. And again, we got these bacillus that kind of are round, but they're kind of rod. Again, just very different. They're in twos, they're diplo, they're in a line, they're strepto, but then these are called palisades, if they're kind of like put together like columns. Again, so what do they look like? The spirillum bacteria are pretty interesting underneath the microscope, and if you get a chance, you can look at some of them under 1,000x. They're very interesting. Very interesting. A lot of them look like a corkscrew kind of thing, like a spirochete or a spirillum. It kind of like float, spiral, or they kind of look like this cholera one, which is interesting. Okay, so again, what do they look like? A lot of times, because of what they look like, we know what the bacteria is, and then a lot of times we really don't know. We have to do more testing, and so what comes along with the more testing is with the cell wall. So here's all the different ones that you can see. Okay. These, as you look, you see all the different packages, and again, some of them, right off the bat, we know that this is the Neisseria group. They look like coffee beans. Right off the bat, we know they're the Vibrillo group there because they look like that. Okay, right off the bat, this is the Mycobacterium group because they look like that, or the Corneae bacteria group, which look like this. Okay, so we know that that's the staph group or the strep group. Again, we know that, but sometimes we don't necessarily know what individual species that happens to be. Okay, and that's where gram positive and gram negative come in. This, of course, is by Hans Christian Gram, and this allows for organisms to be seen better, to stain them. And guys, we did this. We stained our cheek cells with methyl blue so we could see our cells. We stained our onion cells with iodine so we could see the leucoplast, the places where they store the starch. And so here we've got gram negative and gram positive. So the gram negative guys have like an extra outer membrane. So the gram positive guys don't. And so when you stain them, the gram positive guys pick up the first stain, and the gram negative guys are going to pick up the second stain. And so when you do that, you can see a gram negative guys, the second stain is a pink and a red color, and the first stain is kind of a purple color. Okay, and so again, very distinguishable. And a lot of times this will tell you what your bacteria is. Definitely. A lot of times this will tell you which one it is. There are just very distinctive things about them. I remember watching, there was a show, I forget what the name of it was, but it was talking about the bubonic plague breaking out, and when did the autopsy on the person, anyway, bubonic plague causes your lymph nodes to swell, so under your arms you'll get these big sacks of purple. Anyway, it'll bust and it'll be purple looking, and so that's called the black plague. It's why people would look black, kind of like. Anyway, if you see it, you know it, and if when they were identifying it, they were like, I don't know about this, but then when they tested the bacteria and did the gram staining, it's like, this is I think it's a gram negative rod pneumonia or something like that. Anyway, don't quote me on that part, but anyway, that next part said, oh, this has got to be bubonic plague. This can't be anything else. And immediately they double gloved, and immediately they put the mask on, which, by the way, if it was airborne by then, they already got it. Bubonic plague was highly contagious in the airborne, once it reached the airborne stage. Otherwise, it was direct contact. You had to come in contact with the blood, or you had to, a flea had to bite you, or some along that line for you to get it. So again, and then sometimes you get here and you go, I'm still really not sure, and so then there are other tests that you do. We'll talk about some of those in just a minute. The other thing, so here's some gram-positive bacteria, and here's some gram-negative bacteria. So the heliopori is the spiral kind of bacteria that it causes ulcers and is carried in water. And so we see that one. Um, uh, E. coli is a gram-negative rod bacteria, and E. coli can be good, and E. coli can be bad. It can cause food poisoning for us. And so here's anthrax, and so you can't really say that, hey, these guys over here are worse than these guys over here. No, it doesn't fall that way. Okay, and again, here's Clostridium botulism. We're talking about that's food poisoning. So the cholera is right here, and that's food poisoning, and this is food poisoning, and this is meningitis, and this is an STD. So as we look at them, you can't say these guys are worse than these guys. Here's diphtheria. When you get your, I forget, it's called a Tdap shot now, but when I had a shot, it was called, anyway, it's Tdap now, but the D in this is for diphtheria, and the T is for tetanus, and we'll talk about tetanus, diphtheria, and whooping cough in a little bit with the bacteria. So again, this just kind of narrows it down a little bit more. Here, if you want to learn about some of the different bacteria, you can click on the links from the PowerPoint presentation. And again, we can't say that if you're positive, you're a bad guy, and if you're negative, you're a good guy, and all the bad guys are negative, and all the, you can't say that. They're just, again, they're very much opportunists. Bacteria are very opportunist. If given a chance, they'll cause an infection. Yeah, pneumonia, the plague is gram-negative. So I was right. That gram-negative pneumonia would be the plague, and the buboes would be a sign that you have. I don't know if it'll go the plague with real quick for us, but there it goes, maybe for us. Hopefully. So there. So the plague is called Yercenia Pestis. And actually Yercenia is one of the apprentices, proteges of Louis Pasteur. So give you a little tidbit there. So again, symptoms of the plague, you get it can mimic a lot of regular sickness, and there's a buboes underneath the arm. You can kind of see that. And again, this was one of the prevalent ones back in the Byzantine Empire. It wiped out almost, I think, a third of that population. It's been considered, like in this one's talking about, a considered biological warfare weapon. It is, at the time, I don't know if I can find on here, but you can look. At the time, like if it's in the pneumonic form, it's highly contagious. So it's like an 80% killer. But for the most of the time, you get it from fleas, from a rodent. Rats, mice, squirrels, prairie dogs. We don't have it's the black rat that's the main one that passes it. And so you get bit by the flea, and it's a blood disease. So you really got to get it. It's a bloodborne pathogen, but can be a pneumonic one. If you cough it up, then you'll see it. And this is the one pretty much that they, ring around the rosies, ashes to ashes, dust to dust, we all fall down. That's where this one had its roots. Okay, that's where those things come from. So you can look at the different infections just as you go through. Lots of them are real interesting. Cat scratch disease is the one when a cat scratches you or dog scratches you. It's the bacteria underneath their nails, and you want to make sure that you wash the wound. Most of the time, if you clean the wound, you're fine in these. So again, some bacteria actually move, and some don't move. So again, some of them slide and glide, and here's some of the examples here. You can see. It's up to you. If we were doing microbiology, you would have to know that, and you have to stain them a certain way to be able to actually see them, see the flagella with that. The other way, well, let's see. Let's talk about um, identifying bacteria. Do that on your own. Let me see how much time we got here. Come down, little time button. My time button didn't come down. So anyway, um, let's go ahead and answer these questions. Okay, so let's go ahead and answer these questions, and then we'll call this section and end this section, and then we'll get into the next section. The next section, like we talked about, if you can't narrow it down by your gram staining or your motility, then you're going to look at how do they produce their food, and so we'll look at the next section, and then that kind of helps us to identify specifically bacteria. So again, in what major was it what was the archaebacteria different? What major ways are archaebacteria different from regular bacteria? Again, those are the extreme guys. Remember, they live in these extreme environments. They got the little extra piece. They not extra piece, but their cell wall is different, so they don't. They won't. Antibiotics won't work on them. So luckily, we don't really have a chance to come in contact with too many of them. How do scientists identify bacteria? Again, if you go back, they identify them by their shape, how they move, and their cell wall. Okay. What are the three major cell shapes? Again, the cocci, the bacillus, the spiral. You got a bacterial colony is called Staphylococcus bacillus. What shape of bacteria are they possess? Bacillus mean they're the rods. Staph means they're growing in a colony, a group of like grapes, a cluster of grapes. So what type of colony would you see a cluster of grapes under the microscope? So hopefully that helps a little bit for you guys. I hope you got a little bit out of this. Again, if you want to know more about the bacteria, I'll try to put the PowerPoint down below in the comments somewhere, or link it back to my website, and you guys can see it. Okay, thanks again.